Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this video tutorial, we want to talk about UV visible spectroscopy. I've got a request from many of my subscribers to make a video on that. Previously, I made a video on IR spectroscopy and NMR spectroscopy. So if you want to know about NMR and IR spectroscopy, you can look at my channel and you'll get videos on that. So let's begin with UV visible spectroscopy. UV visible spectroscopy is very widely used technique in not only chemical or chemistry labs but also in life sciences. Many different experiments rely on the idea of UV visible spectroscopy. But the first question is why we need this technique. If it's a technique, then what this technique is giving us? The answer to that is, in many situations we need to find concentration of different substances, concentration of different molecules in a solution, and that we can measure perfectly with UV visible spectroscopy. And in some cases also concentration of cells during the microbiology practicals, we can also use UV visible spectroscopy to measure. So it can give us a good data about the concentration of any molecule that is present in a solution, but it also can tell us a little bit about the chemical group that is present in the molecule, but the data that we get is not very much reliable, because for the different types of group the UV visible spectroscopy can give us very similar data. So we generally use it for only concentration analysis. But how exactly we will get to know the concentration from the UV visible spectrophotometry? Let's look at that in this video. To understand UV visible spectrometry, we also tag it with known as a colorimetry, which is linked something with the different colors of the color substances, which can help us to identify the concentration of the colored compound that is present in there. And we use it extensively in enzymatic reactions to find out enzyme associated activities and enzyme kinetic studies. So the first thing that I will tell you is what is UV visible spectroscopy and how the whole technique is oriented, like how the mechanism of the technique, so all these instruments that are present there to make this technique possible. Then I'll tell you the molecular mechanism with which this whole technique works. So let's begin with the actual setup of this UV visible spectrometry, and that gives us start with a source that is definitely a light source, because as we see, UV visible spectrometry utilizes UV or visible range of light as the energy source, primary energy source to find out the important feature of concentration. In case of IR spectrometry, we know that infrared is the range of the light that they've used, but in this case we used UV or visible range. And you know, UV and visible range, like electromagnetic range, you should have a little idea about the electromagnetic wavelength that we see completely. And some basic idea before talking into the details is, if you look at the electromagnetic spectrum, you'll find out different wavelengths. It's ranging from gamma rays to ultimately ending there in the microwave as well as the infrared waves till the end. So what those different waves are telling us? The important feature you always need to know is the idea of two things. One is the wavelength and the frequency of that electromagnetic wave. The more the frequency of a wave, the higher energetic that wave is, that energy is. That's the idea. More frequency, more energy, and if you have more frequency, the wavelength will be less. More frequency, less wavelength, higher energy, and less energy means less frequency and longer wavelength. So if very long wavelength, that will give you less frequency and less energy. Higher shorter wavelength, more frequency, more energy. So in this case, if we divide this UV visible spectrum into the small part and fractions, what we'll find here is simply ranging mainly from 200 to 800. That's kind of the range, although we can extend it till 10, 20 nanometer in the wavelength, but from 200 nanometer to 800 nanometer, that will be the range. From 200 to 400 approximately taken as a UV range, and from 400 to 800 approximately taken here for the visible light range that we usually use for the UV visible spectrometry. But normally actually UV range ranges even back less wavelength, less number of nanometer of the wavelength till this part as well. So what we know here by looking at this spectrum is that as we are going close to UV, the wavelength is shorter, so the frequency is higher. and as we're going this side the frequency is lower. So this right hand side we'll find more like a microwave, IR or infrared, that means long wavelengths, less energy, less frequency, till we go towards UV, X-ray, gamma ray, that means very short wavelength and frequency is very high, energy also very high. This is the scheme. Now from this scheme we only take the 200 nanometer to 800 nanometer range. Now in this case, whenever we go to 400 to 800 nanometer, we have the visible range of the light, that is visible colors that we know, the VIV or the color violet, ultraviolet, that from the violet till the red, that is the different 7 separate colors arranging from this part of the visible range. Now what we do now is simply, if we look at the instruments, it goes like the source. Okay, so like any kind of light source, let's say filament in there, this is the light source, and after the light source it has a separate unit known as a monochromator. Let me draw this unit. This is monochromator. What monochromator does is, normally the source light, as it will have visible and UV range, it have this basic light that we know, the light coming from the light bulb and the headlight of your scooter. So that thing contains 7 separate, all these different wavelengths. So monochromator separates all those different wavelengths of the light into single beam. That's why called monochromator, because it will make single beam from all this mixture of this light. And after the monochromator, what else we have? We have the beam separator, and the beam separator almost like almost what we can say, a simple knob which can separate the beam and guide it into 2 separate directions. And in this 2 separate direction we have 2 separate chambers. One is for the control and the sample chamber, and then right after both these chambers are connected to a detector. This is the simplest drawing of any UV visible spectrophotometer, and it begins with the source, and the source light comes in, and here we have small gaps, slits, through which the light passes. After passing there will be this mirrors will reflect it, but ultimately there is a prism inside this monochromator. So there is this prism. So as this prism will separate that light into all these different types, all this different wavelength of the light, different colors. So only one specific wavelength will be allowed to pass through this ultimate chain. Now you can alter this, like anytime it can pass the red light, it can pass blue, it can pass orange, it can pass yellow. It depends on in which angle the channel is present, through which it will go to this beam separator, and this beam separator will separate the same monochromatic light, send it to 2 chambers, the chambers with the control and the chambers with the sample, and then from both this they will get the data by the detector, detected by the detector, and then finally they will record the data and give you the answer to it. Now the question is, in the simplest structure that we see, why this kind of structure is prepared and what exactly our role in this case. The idea of UV visible spectrometry is simply, whenever the light hits, a specific wavelength of light hits a molecule, that molecule get excited, and the electron, once excited, is jumped up from a lower, or we can say ground state, to a higher energy state. And whenever the electron jumps out, what it does, it absorbs that particular light energy, because the electron utilizes that energy to move from the ground state to the top state, right? Because one thing all your life is that energy cannot be created, it cannot be destroyed. The only thing possible is transforming energy from one form to the other. So in this case, getting the energy, so energy is little bit absorbed at that point, energy is utilized at this point in a more scientific way. So what we know, if the energy is utilized, then what we know, the intensity of that light that received is lost a little bit, right? So what we know, if we put a blank, let's say cuvette are small chambers where we add samples to test. So generally they are made up with glass. So this glass cuvette is present here, and in this cuvette we only put water. So there is no molecules present there, nothing is there, it's completely basic water. So what happens, that light beam passing through water, and water, there is no molecule to finally absorb any light or utilizing the energy from the light. So what it does, simply it usually passing almost the same intensity that ultimately reaches here. So what we get here, there are two separate intensity that a detector always can measure. So in both this cuvette we put water, then what will happen? Same light that will come. So normally the light that generally come, if there is no absorption, known as the intensity of the light, starts with I0. This is the start point of the intensity of the light. And then if we put the sample, then we get another intensity depending upon the molecule that is present in that sample. But here same, so we also call it I, because that's the intensity of the sample. But in this case we know, if we put water in both the chambers, then we'll get the value of I0 will be equal to I, will be same. So the intensity of start and intensity at the end are same. That means we're dealing with same solution, and literally this solution is not taking any intensity that is first transmitted through this beam separator. But now imagine in the sample tube, in the sample chamber we put a cuvette, and here what we put a solution, let's say protein. Let's say we put protein solution in there. Now proteins, mainly there are bulkier rings in the proteins and all those aromatic amino acids. Those amino acids will receive the energy at a specific wavelength. And if you also put DNA, they will also absorb a specific wavelength of light compared to the other. And that is also due to the bases that are present in the DNA. So usually for proteins, that we know that it will absorb the 260 nanometer of the wavelength. So once they receive the 260 nanometer of the wavelength here, and that causes absorption of that wavelength, absorption of few of this energy by the number of protein molecules that are present there, the amount of protein, the concentration of protein that is present there. The more protein present there, the more absorption, and the less is this intensity of the incident and intensity of the reflected I, because light is passing, reflected, and finally reaching the detector. So the more absorption there, the I value will be decreased. So in that case we know there is a net change in intensity, and what we can find about the net change is this is the net change in the intensity, isn't it? So what we know here as known as transmittance. Transmittance known as the value for, so we call it transmittance, or capital T, the value for I by I0. That is known as the transmittance. So the more light is refracted, the more light is removed. That means the more transmission take place. And the less light is removed, which is the value of I will be less, the less transmittance is there. So if we know the value of transmittance, now what is about absorbance? Transmittance and absorbance are opposite to each other, different to each other. So if transmittance is I by I0, in that case absorbance will become a log value of I0 by I. That's how you measure absorbance. So more transmittance, less absorbance, less transmittance, more absorbance, and that's kind of a true. And it depends on the concentration of the molecule that is present in the sample. Now what kind of molecule that is present there dictates also what kind of group, chemical group is present there, also dictates at which wavelength they will receive the energy. That is also very, very important. And for every single type of chemical groups, there is a specific wavelength for which the absorbance is maximum, known as lambda max or lambda maximum. Lambda max is the maximum wavelength for which they will give you most absorbance, and the readings there known as lambda max. And we can also get the value of lambda max for different types of chemical groups. Now the question is why they absorb this light and how this process works molecular level. We will talk about that in a moment. But before going in there, let me first tell you how exactly we interpret this data, because detector detects two things, I0 and I. The difference between the two will tell you whether they'll absorb some light or they transmitted more light. So for that, what we can find in this particular case is simply we can plot a data with the absorbance and the difference in the absorbance by the percent, like the difference is the absorbance along with the concentration. So if you know if you know the absorbance, we can also find the concentration from there. But how exactly? Because you know the transmittance, if you look normally in this data, they are not going to give us absorbance as a value, because the detector detects the transmittance, right? Because absorbance is there in the sample and detector only detects how much is transmitted, how much transfers through this cuvette to the detector. So always the value that we'll get will be in the transmittance form. So simply what we can draw from here, we can draw a graph, and in that graph what we say the percent transmittance and also the concentration of the solution of the molecule. So transmittance will be almost the value of 10 to the power minus concentration into the length of the cuvette, that is the length of the cuvette dictates also how many molecules of that molecule is present in that cuvette. So according to Beer and Lambert, they told us that T or transmittance can be measured in the form of 10 to the power minus concentration into the length. But transmittance is not going to give us much data, because if we put the data in the terms of transmittance, let's say the concentration is very less, transmission should be maximum, right? Absorbance in that case will be very less, so transmittance will be highest. Then let's say a little bit, then a little bit more and more and more. So what kind of graph we'll get? We'll get a graph like this, a slope like this. This is not a straight line. This is not a linear graph. Now the problem with this graph is, although we are getting the data directly from the detector as a percent transmittance and the concentration, it will be very difficult for us to extrapolate the data. Let's say we know samples and we utilize some test samples, the concentration which we know, and we plotted the plot with it. Then what we do, then we have an unknown and we're testing that unknown, and somehow the unknown is near here, so it will become very difficult for us to finally calculate what exactly the concentration of that unknown. It becomes really difficult. But if the data is always straight line, it becomes very easy to predict the concentration of any unknown sample, and that's exactly what we do. Normally in lab, if we are checking up a protein concentration, we first prepare standard concentration of proteins, that the concentration of which we already know, and then what we do, then we plot a data with it, a straight line, and then the unknown sample, whenever we get the absorbance, by putting at the data, we can simply extrapolate the data to the concentration to find the actual concentration of that protein. But how exactly we get a straight line in the curve? That is a big problem, and the only way it's possible if we get the data in absorbance scale, because no, if in absorbance scale the data should be something like that. It will start from low concentration, low absorbance, high concentration, high absorbance, a linear relation. But the transmittance is not a linear relation. So in this case, to make it a linear relation of any curved line, remember that in your lifetime you will always do a log. So the log value of concentration into length, that can give you the value of absorbance. That will give you the value of absorbance. And the log value that we utilize here is simply utilizing as a constant that is known as molar absorption coefficient, or epsilon, into concentration into length. We also can write it as epsilon, concentration is written as C, length of the cuvette is known as L. So the formula comes down to epsilon CL equals absorbance. This is the net absorbance formula, ECL, derived from Lambert-Beer equation. And from this equation, what you can also tell, if you know absorbance, because in the experiments we can find absorbance. So if you know absorbance, you can find concentration. So for a concentration, it will be absorbance divided by epsilon into L. This is how we can calculate a concentration of an unknown molecule that can be protein, DNA, or any other molecule out there. By this fashion, what we can create a straight line of the graph, and we can kind of easily predict the concentration. Let's say this is the straight line of the graph, and we know the unknown protein is somehow represented and present someplace like here. So the concentration is simply drag, and we'll find it here. And in that case, instead of transmittance, it will be absorbance. So that's how we'll get the concentration value from the graph as well. Okay, so this in a sense is the mechanism of how we use UV visible spectrometry and to get the data out of it. But the second part is also left. Why and how it's working in the molecular level. Remember, for all these type of molecules, I told you, like for all these different varieties of molecules, there is only a specific wavelength for which that molecule will get some energy and absorb the energy and do some transmittance of the energy. Now, what is that mechanism? To understand that, let me erase some of this. So what we know at this particular point is all the different molecules, they have chemical groups, like different bondings inside. So for any molecule, if we look at here, it's simply we can divide in their energy states, and energy states with the the ground states, like the non-bonding state or N, and then we have pi star, sigma star, sigma. This is almost like the simplest idea of how the energy states looks like in the atom. So in this particular case, we have the sigma, pi, N, which is a non-bonding, and then pi star and sigma star. These are also non-bonding, but these are the bonding. So the idea is we have carbon. This is a single bond present between carbon and carbon. This is a pi bond. So what happens normally here in this case of carbon and carbon, this is sigma bond. Very sorry. So if it's a sigma bond, strong, very much strong sigma bond, and sigma bonds require more energy to create and break. So here sigma bond is there. So the electron that is present, let's say here in this orbital here, and the electron, whenever they receives a specific energy, it can jump up to the sigma star. So it will jump from one orbital to the other. This is the ground state to the higher energy state. But the problem is, in the higher energy state, the bond will not be very much stable. So again, it needs to be moved from that. So this is how the electrons jumped from a ground state to a higher energy state. For a sigma bond, directly from sigma to sigma star. Now, they can also jump to this non-bonding orbital, but it will, if it takes only less energy. Similarly, if it's C double bond O, in that case we know this is a pi bond. So for a pi bond, the journey will begin from this pi and it will go to the pi star, and from the pi to the N. Now it depends on how much energy available and what is the exact wavelength of light that's hitting. That's why they rely on a specific wavelength for excitation. For UV visible spectrometry, it's always the excitation of that molecule in response to the energy that they receive, in response to the wavelength of light that's hitting. Now, if the wavelength of light is shorter, that means very much high frequency, high energy, for a sigma bond, it will directly jump to the sigma star. But for a pi bond, if it's work for a very comparatively less energy, it cannot push the electron to the pi, from pi to pi star. Instead, it will push from pi to the non-bonding orbital. And that's how the whole thing works. And based on that, the absorbance also vary, because the energy taken for the electron to jump from the ground state to the energy state is lost. That's the energy which is absorbed. Rest of the energy is transmitted out. That's how we find the transmittance. Okay, so in a turn, this is the idea. From looking at this idea, you can tell we kind of can be enabled to get an idea about what kind of bonds that are present and what kind of chemical groups that are present in a molecule. But in reality, there are so many molecules with similar types of bonds, and the same formula of those molecules you cannot recognize. We cannot recognize the exact group by looking at this technique. That's why we don't use it to get an idea about the specific chemical bonds that are present, which we'll use for NMR, which we'll use for a magnetic resonance spectroscopy, as well as in case of IR spectroscopy, but not in case of UV visible. We only keep UV visible for the process of concentration analysis of molecules. Okay, so that's it. If you like this video, please hit the like button, share this video with your friends, and subscribe to my channel to get more and more videos like that. Thank you.